Thank you so much for that wonderful introduction, and I'm glad to see so many people here and so much interest. So today, I will talk about mitochondrial dysfunction in autism and the roads to understanding it and treating the mitochondrial dysfunction to improve symptoms in autism. Just some disclosures. I have funding from many places, including the Brain Foundation, and also are doing industry clinical trials in our advisory boards. I'll just say a quick disclaimer I have in every talk is that the treatments that we talk about many times, and for the most part for autism, are not FDA approved treatments. That doesn't mean that they can't be used, but it means that we have limited evidence at this time, and that it's important when using treatments to consult a medical professional who's knowledgeable about the risks and benefits of any type of treatment that you would use, both off-label and on-label. So, today we're going to talk about the mitochondria. The mitochondria is a very important part of almost every cell in your body. It's best known for making energy. You have anywhere from hundreds to tens of thousands of mitochondria in each cell of your body. And it's a very complex part of the cell. It actually has two membranes to enclose the machinery. It has these little folds in the inner membrane called cristae, where a lot of the work happens. It actually has some of its own DNA. It's the only other part of the cell that has its own DNA besides the nucleus. So it's very complicated because some of its functions are regulated by the genes that are specific to the mitochondria and not on the chromosomes. So we have our normal mitochondria, which has lots of power, gives us energy. And then what we're thinking is that sometimes there's dysfunctional mitochondria where it doesn't seem to be working that well. It doesn't have the power to do what it needs to do. And what we're learning is that this is not just a circumscribed, what we call mitochondrial disease, but that the mitochondria, being central to many of the cellular processes, is actually dysfunctional in many disorders, since it's so important to support cellular function. Really, we're still learning about mitochondrial disease and dysfunction. Mitochondrial disease really was first described in 1988. The first papers were published on the first mitochondrial diseases in Science and Nature, two really good journals, of course. And so when we think about it, in the time span of medicine, 1988 is pretty much a blink of the eye. So that's why we're still learning about mitochondrial disease and dysfunction. And one thing we know is that when there is a problem with the mitochondria, we see that there's symptoms within the high energy dependent tissues of the body. And these include most usually the nervous system, so neurologic disease, the GI system, so gastrointestinal disease, and the immune system. And when you think of the comorbidities that we see in kids with autism, these are the three areas of the body that are most affected. So it's no wonder why the mitochondria seems to be involved possibly in autism. And the other important thing to know is that the mitochondria is not just the powerhouse of the cell, but it's important for other functions, including regulating cell death and oxygen radical regulation. Really, the evidence for mitochondrial disease and dysfunction is growing. And probably, I think one of the best reviews was on the paper that Dr. Rosignol and I published in 2012. We were kind of interested. We had heard that there's more and more evidence that there might be some involvement of the mitochondria with autism. So we did something called a systematic review and meta-analysis to see what the evidence was. And we found some very interesting findings. Our findings were that the papers that were published, there was 3 papers to look at very strictly defined mitochondrial disease, which is a very particular specific diagnosis. And we found that studies that looked at that found that about 5% of children with autism have mitochondrial disease, which is a very severe form of mitochondrial dysfunction. But when we looked at studies that looked at biomarkers that the mitochondria wasn't working, that is elevated lactate or pyruvate or lactate to pyruvate ratio and such, we found those rates were much higher, somewhere around 10 to 30%. And so what that suggested to us is that there was something going on with the mitochondria, that is, the mitochondria wasn't working, but it may not fall into that category of mitochondrial disease. So there might have been this broader sense of mitochondrial dysfunction. And so, and that's not really, surprising in a lot of ways, because one of the things we know is that the mitochondria is affected by environmental factors, both the external environment and the internal environment. So the external environment, like toxins, like the microbiome, the microbiome technically is outside your body, even though it's in your gut, but also things internally. Oxidative stress in the body and inflammation will affect how the mitochondria functions. And we know these are all involved in autism. So it's not surprising that the mitochondria would be affected. But it's important to know that when the mitochondria isn't working, there's consequences, including increased oxidative stress and inflammation, autoimmune disease, immunodeficiency, neuronal dysfunction, problems with neuroplasticity, cerebral folate dysfunction, autonomic dysfunction, and gastrointestinal dysfunction. And what's important about the mitochondria is, again, it's at the center of many of the important cellular processes and many diseases. And it can, when it's not functioning correctly, it can start a vicious cycle, which can cause more dysfunction. And this is just one diagram that shows, well, dysfunctional mitochondria can increase oxidative stress, which can cause inflammation, which can cause more mitochondrial dysfunction. It can also deplete glutathione, which is important for mitochondria to work, or actually create mutations in mitochondrial DNA. So the mitochondria may be very central to a lot of the physiological abnormalities we see in autism. This is one paper we've published recently. This is a diagram from there where we looked at the prenatal factors that could affect the mitochondria. And here you see on the left a list of prenatal factors that have been linked to autism, and how they either negatively, that is in orange, or positively, which is in green, can affect mitochondrial function. So there's a lot of evidence that many of the prenatal factors that are linked to autism could act by affecting the mitochondria prenatally. And we have some evidence of that also. And of course, on the right, we see mitochondria is important for many important pathways in the cell. I'm not going to be able to talk about the evidence that we found in the papers that we've published, but suffice it to say, we have a series of papers where we've looked at prenatal environmental influences, both air pollution, prenatal exposure to air pollution, and prenatal exposure to metals, both nutritional and toxic metals, to see how that affects long-term mitochondrial function. And we were actually able to show that that the mitochondria is modulated long-term, that in childhood you can actually measure mitochondrial function and link it to those prenatal exposures, suggesting those prenatal exposures may have actually formed the physiology of the mitochondria, have influenced the physiology of the mitochondria early on, and that these differences, these changes, were different between those that have regressive autism and non-regressive autism. The other thing that we have been able to show is that indeed there is a unique type of mitochondrial dysfunction in autism, particularly those with neurodevelopmental regression. And this is a recent paper we published with Andy Zimmerman, who was originally at Kennedy Krieger and then Harvard and UMass, where we were able to take samples from patients that he studied with and without neurodevelopmental regression with autism, to show that those that have neurodevelopmental regression in autism actually have very active mitochondria that where their respiratory rates are much higher than normal. And this is important because usually when we think of mitochondrial disease, we think the mitochondria is not working well, so the respiratory rates are very low. Here we've actually linked a subgroup of autism to extremely high respiratory rates of mitochondria. So we'll talk about some of the grants from the Brain Foundation that we've received that is helping us find treatments and learn more about the mitochondria. This is one grant where we've tried to just simply take a mitochondrial supplement that should improve mitochondrial function, and very critically look at it through a double-blind placebo-controlled clinical trial. So there's a controversy about whether you should treat mitochondrial dysfunction or whether it has to be mitochondrial disease, whether you need a particular diagnosis of mitochondrial disease. And this is a nice paper with my colleagues Dmitry Nizov and Steve Kaler, where we argue and we give vignettes of diseases where you see actually mitochondrial dysfunction secondarily. And it's clearly not a disease of the mitochondria, but the mitochondria is affected. And we discuss how treating and to support the mitochondria, even though it's not the primary part of the disease or the primary cause of the disease, can actually improve function. How do you treat the mitochondria? Well, there's many different ways, but suffice it to say there are certain supports with certain specific vitamins, many of them B vitamins, but also CoQ10, creatine, certain folates, and support for oxidative stress. I'm not going to go through them all. But there are certain vitamins, and in our study, we use the NeuroNeeds vitamin, which is actually designed to support mitochondrial function, along with the Q-Needs vitamin, which is a vitamin kind of cocktail to support mitochondrial function. We developed a double-blind placebo-controlled crossover trial where first we enter individuals that have some type of mitochondrial dysfunction as measured by non-invasively with the buccal swab technique, and then treat them with either the actual supplement or placebo, and measure how their mitochondrial function changes and how their symptoms change over time. This is an FDA regulated trial, so we asked the FDA and filed for an IND not to approve this as a drug at this time, but to increase kind of the level and of quality of the trial. And we had two aims. Our primary aim was to determine if this supplement could improve mitochondrial activity in 50 individuals with autism and abnormalities in mitochondrial function, and two, to see if it improved the core and associated autism symptoms. So what came out of it? Well, all these things are a process and a learning experience. So, of course, getting FDA approval, one of the things that we found is that we needed a certificate of analysis of the investigational product. And when you have a combination product like this, and this is why I don't think drug companies like to make combinational products, it's very tough to actually investigate analyze the product once it's mixed. And there is a lot of different issues and certain chemicals interfere with other chemicals, certain substances interfere with others when you're measuring these things. So it took us about a year to find a company that could do this with enough precision and within a reasonable price point, because we had price quotes anywhere from $5,000 to $2.5 million. And we were able to do that, and then we had to produce a placebo that tasted identical to the product, and that took about a year. We had to do taste testing and blinded taste testing to actually develop the product. Of course, we had to advertise and recruit for the study, and then we had to find the participants, which is a little bit slower than we expected, which I'll discuss in a second. And then one of the major issues we found is the tolerating of the treatment, and it's about 60%. And I don't think this, in our experience, this isn't really specific to this product, but to treatments in general in kiddos with autism because of their particular aversions to certain tastes and textures, and really tells us that some of the ways that we've delivered products as far as pills and powders and such, and liquids and such, may not be optimal, and we have to, there's probably a whole area of research we have to look into of how to develop products with better delivery systems. We in the study we had 29 that were eligible, and in order to be eligible, after in screening, you need to actually have confirmed mitochondrial dysfunction. And unfortunately, we were hoping to get those tests back with a 2-week turnaround time, and it's been a 3-month turnaround time. So that slowed down entering people into the study. And we've lost a couple to follow up because they got tired of waiting. So it's new technology, and it's unfortunate that it takes time, but we're moving ahead, and we now have 3 completed, but we have definitely some that are going through that are in part 1 and part 2, and others waiting to enter the trial. So taking a little bit longer than we had expected, but I think we're learning a lot, and we'll get some really good information from it. The second, approach we're taking is by looking at in vitro cellular models. We started out this research many years ago using lymphoblastoid cell lines, and now we've moved into using fibroblasts to confirm and further our studies that we had in the lymphoblastoid cell lines. We use something called the seahorse. The seahorse is a instrument that has really revolutionized looking at mitochondrial function. It used to be that you could only measure 1 or 2 samples at a time of mitochondrial function. You had to isolate the mitochondria, which is a bit of a chore. And so the seahorse was developed about 10 years ago, and it allows you to measure mitochondrial function in living tissue, so in cell culture, in a 96-well plate. So you can put in replicates, you can manipulate the cell lines, and you can put both control and your experimental cell line on the same plate, which gives you a very high throughput and allows you to measure a lot of things. And so one of the manipulations that we decided to do when we started this was to see how, not just how the mitochondria was working, but how the mitochondria was resilient to stressors, particularly oxidative stress. So we developed an assay where we can use something called DMNQ, which increases superoxide in the cell, so that we can see how the mitochondria can actually handle stress. From this assay, we measure several measures of mitochondrial respiration, including what we call ATP linked respiration, which measures how much ATP or energy the mitochondria is making, proton linked respiration, which is very important. And this is actually a type of respiration that controls oxidative stress. And it does not make ATP or any energy. So it decreases the efficiency of the mitochondria. And then we can measure what we call the maximal respiratory capacity, how much the mitochondria can function, how high its respiratory rate can go. And then I take this difference between the basal respiration and the maximal respiration, which tells us about the reserve of the mitochondria, how much reserve the mitochondria has if it has to actually increase its respiratory rates due to stress. We verified, so again, we did this assay where we used DMNQ to cause stress, and this is just an assay where we verified that actually the mitochondria was being stressed. The red color you can see, which progressively gets more and more as we add more DMNQ, is a mitosox red, which measures mitochondrial superoxide, so oxidative stress within the mitochondria, to verify that our assay is doing what we think it's doing. We've done several experiments on what we call lymphoblastoid cell lines, which are immune cells that have been immortalized. And what we showed, and this is one study that looked at patients with autism, and they're typically developing siblings as well as unrelated controls. And what we did show is that the respiratory rates were much higher in the kiddos with autism as compared to their typically developing siblings and the unrelated controls. And one thing we did show that's not in this slide is that the siblings tend to have high levels of oxidative stress also in their cells. So the thing that differentiates the siblings and the kids with ASD or is the abnormal mitochondrial function. We also showed that if you divided up the kiddos with autism into those that had worse and better repetitive behaviors when these cell lines were harvested, or when the blood was actually taken from the child several years before these studies were done, that we could actually differentiate which children were more severe by measuring their mitochondrial function. In larger samples of lymphoblastoid cell lines, what we've been able to do is show that there's these two subgroups. There's a subgroup of children with autism who have normal mitochondrial function, and then there's a subgroup that have these very high respiratory rates, which I had previously shown you. We've linked to neurodevelopmental regression. And what we showed was that not only do they have these very high respiratory rates at baseline, but when you stress these cells with DMNQ, these respiratory rates plummet much faster in the kids with autism than in the controls, particularly those that have these high respiratory rates at baseline, suggesting that there's this subgroup of kids with autism with a unique type of mitochondrial dysfunction who are very vulnerable to physiological stressors. And we've looked at some of the reasons why molecularly. In one study, we were able to link this to a protein called UCP2, which regulates oxidative stress at the inner mitochondrial membrane, and showed that this protein was upregulated in the subset of lymphoblastoid cell lines from the kids with high respiratory rates and autism. We've also linked this to changes in gene regulation, where we've found that the kiddos with high respiratory rates fail to upregulate important genes that combat stress, physiological stress in the cell, including PGC1 alpha, PINK1, MFN2, SIRT3, SIRT1, and so on. We were also showed that this might be related to regulation of mTOR, and that low-dose rapamycin may be able to correct some of these changes. And in one in vitro study, we showed that by using low-dose rapamycin, we could actually normalize the respiratory rates of these cell lines. So now we're actually moving on from the lymphoid blastoid cell lines to fibroblasts to confirm these same findings in a different tissue, to make sure that this isn't, just something because of something particular to lymphoblastoid cell lines. And then we're taking this a little further. And in this study, which was a collaboration with the French, we were able to use confocal microscopy to look at mitochondrial morphology, and actually showing that the shape of the mitochondria is different in cell lines that have these high respiratory rates versus the normal respiratory rates. That is that the cell lines with high respiratory rates had more complex mitochondria that were clustered together and closer to the nucleus, whereas the ones with normal respiratory rates had much longer, more evenly distributed mitochondria in the cell. And what we're doing now with the grant from the Brain Foundation is taking treatments that have been shown to be therapeutic in autism and other neurodevelopmental disorders that also can normalize some of the gene targets that we found, and systematically go through to see if these will normalize the respiratory rates of the cells and normalize gene function. And so far in the fibroblasts, we've done rapamycin, metformin, NCAR we're doing right now, and we have several others that we will do in the near future. The next study is very exciting, that we were very lucky that the Brain Foundation supported this. We have the very unique opportunity, since we are in a hospital setting, to actually have access to fresh excised brain tissue in order to look at this tissue and measure mitochondrial function in fresh human brain tissue. And this has never been done before. So of course, this has been done in animals, in animal models, but never in humans. And so we have really a unique opportunity to look at mitochondrial function in brain tissue, not just theoretically or through imaging, but actually, using the seahorse instrument. And so, of course, the individuals that have brain removed have that for a reason, and usually it's for epilepsy. And we have children with epilepsy who do and do not have autism. So we can actually look at mitochondrial dysfunction, the changes associated with epilepsy, changes associated with autism, and correlating these with other biomarkers like high frequency oscillations, which we believe may be linked to a unique type of mitochondrial dysfunction. So we're characterizing mitochondrial function in those with focal seizures and other brain tissue. We also can correlate this with electrophysiological measurements and genetic changes, RNA expression and transcriptomics, and also correlate this with high frequency oscillations. At this point, we've actually received tissue from different parts of the cortex, including the temporal, frontal, and occipital lobes, deep brain structures, including the hippocampus, parahippocampus, amygdala, and various pathologies, including cortical dysplasias and actually tumors too. So we are looking at some cancers. We've adapted a protocol that's developed by one of the scientists in our lab, who developed this on animals, on rodents, when he was at University of Kentucky. And it not only allows us to isolate mitochondria, but also to divide into what we call synaptic and non-synaptic samples. We've adapted the seahorse protocols so that we can measure various aspects of the mitochondria and its respiratory rates. I'm not going to go into the details. Again, measuring both synaptic, non-synaptic mitochondrial function, doing genetic analysis, looking at mitochondrial morphology using electron microscopy, and also being able to localize these things in the brain. This is a picture of these high frequency oscillations, which we believe may be specific to seizure focus. And we believe that in order to have those high frequencies and mount neuron or energized neurons to have these high frequencies, you need very high respiratory rates in the mitochondria. So we're able to correlate this with this high frequency oscillations by depth electrodes, which we see here, also with MEG, and we also do resting state functional MRI and SPECT on many of these patients. And these are just some of the results. What we did find indeed was that the seizure focus had both complex 1 and complex 2 respiratory rates that were much higher than the non-focal part of the tissue, the part of the tissue that was not the epilepsy focus. So using that as a control, showing that the epilepsy focus had very high respiratory rates. And we were able to see the difference between synaptic and non-synaptic mitochondria, and finding that the difference tends to be in maximal respiration. We're also coordinating and collaborating with NIH and their electron microscopy lab at the Cancer Institute to also look at changes in mitochondrial morphology with electron microscopy. And here's a nice shot where we can see that they're the border of the seizure zone. The mitochondria on the left is a little bit more intact than the mitochondria on the right, which is in the seizure zone, which is very swollen. The cristae, those folds, you can see are very swollen and very enlarged in those mitochondria on the right. So with that, I thank you for your attention, and I will take any questions.
There are questions in the Q&A section.
Okay. Super. So which is therapy which resolves this problem lifelong? Possibly. I mean, of course, we always try and use therapies that are going to be very safe and well tolerated. So they could be, if they needed to be lifelong or for prolonged periods of time, they could be. But the great thing about the mitochondria and what we hope is that the mitochondria is what we call plastic. There's what we call mitoplasticity. And we think that it may have gotten into this abnormal state because it is trying to account for certain stressors. And if we take away those stressors, maybe the mitochondria will help repair itself, and you can decrease the treatments. Can these gene regulation tests be conducted outside of studies? Well, not, most of gene regulation tests are really in the research phases right now. There are, definitely labs that are looking into how to use them as biomarkers. So I think that's in the future. So I don't think that we have those right now as more diagnostic tests. Yes, is the teeth research with Dr. Arona? Yes, most definitely. He processed the teeth for us. We published one paper in Translational Psychiatry, which was extremely interesting. I didn't go into it in detail, but the very interesting thing is that we found that the metals that were important prenatally were more nutritional metals of zinc, manganese, and copper, all which point to superoxide dismutase and control of oxidative stress, we believe, prenatally. But, we need to do more research, and we're trying to get NIH funding to continue that research because, of course, he's done some great research showing that these metals are dysregulated, and we're hoping to take it to the next step to actually show, well, what's the consequence of that prenatal dysregulation of metals. Regressive autism disorders, 16 years old, nonverbal, 5 mitochondrial markers, looks heavy mitochondrial impact. Yes, I mean, definitely the next step would be to actually measure mitochondrial function either with the buccal swab or other techniques, skin biopsy, muscle biopsy, if appropriate. Of course, you need to consult an expert in that field, in the field of mitochondrial function, to decide that, and also genetic testing, whole genome with mitochondrial DNA sequencing, probably. But of course, I'm not giving medical advice. You need to consult an expert, but I would pursue that. Did the kids with worse repetitive behaviors have high? Yes, they did. That's what we found in the lymphoblastoid cell lines that was from the AGREE database. I'm sorry, biorepository. Can these, which there is, if a child has consistently low numbers of mitochondrial swab, do they fall under? No, probably not. I mean, I'd have to look at that, but right, we see that there seems to be 2. I don't want to, we're very excited about these kids with very high respiratory rates because it's unique, but there's many kids with very low respiratory rates that fall into that category of more classic mitochondrial dysfunction who also need aggressive treatment. So definitely important to pursue that with an expert in the field. Can this help with PANS pandas? Yes, most definitely. One of the things, of course, it's important to point out is PANS is not defined as an immune disorder. It is an acute onset of symptoms, and this at least I'll tell you, when I was in residency, anybody with neurodevelopmental regression or changes, acute changes, had mitochondrial issues until otherwise found. So I think that when we look at any acute changes in behavior, that we have to think in a very broad sense, and this is a whole nother talk I have of looking at the differential diagnosis, including metabolic immune disorders, seizure disorders, and such. And we have to really cast a wide net when we look at those patients that we put in that kind of PANDAS category. And mitochondrial dysfunction itself can contribute to immune dysfunction. So at least in my practice, I found that a lot of kids, even with immune dysfunction causing PANDAS, also have a mitochondrial dysfunction, which needs to be treated simultaneously. So it's very complex. Is there a theoretical role for preconception and prenatal mitochondrial testing and supplementation? Most definitely. Most definitely. Is using lactate and pyruvate ratios as a screening tool not be enough? Yes, it's not enough. So lactate and pyruvate are just one measurement. One of the measurements that we find that picks up some of these kiddos, lactate and pyruvate can be normal, but alanine to lysine ratio, which was really developed by Richard Kelly, is important. And of course, we have a paper on dysregulation of fatty acid oxidation pathways and unique changes in acylcarnitines that may be linked to mitochondrial dysfunction. So yes, it's complex, and especially I think in these new types of mitochondrial dysfunction, we're still learning what the biomarkers are. N-acetylcysteine part of the neuroneeds mixture. I'm not sure. I think there is, but I would have to check the list for you. I don't want to misspeak, but the neuroneeds is a pretty good mixture, support mitochondrial function and oxidative stress, and I believe there's some N-acetylcysteine in there. If not, the only bad result on mitoswab is borderline. What does this mean? Again, I think you need to consult an expert on mitochondrial function and looking at the mitoswab to tell you that, because it can be tricky to sort out. Thank you for the presentation. Have you tested and identified ASD individuals who have normal mitochondrial function? Yeah, we wonder a lot of times, are there any with normal mitochondrial function? Because we find so many. Yes, there are patients with normal mitochondrial function. Despite all the tests that we've done, there are a good number of kids that have normal mitochondrial function. So, when we can find it, it's good to treat. But it's not the only thing. I think it's the treatments are very complex. And they have to hit it from many directions. So when we can find that there's mitochondrial dysfunction, I think it's good to treat, but there's a lot of kids with normal mitochondrial function too.
Excuse
So,
me, Professor Frye, I think
yes.
you're out of time.
Okay, sorry, lots of questions. Great, so shall we move on?
Yeah, we'll put up the next bio.